Every living cell stands at the crossroads between survival and decay. Within this constant molecular flux, a quiet force sustains equilibrium, a process known as autophagy. First described in the 1960s under electron microscopes as strange double-membrane vesicles engulfing cytoplasmic fragments, autophagy was once dismissed as a passive consequence of stress. Today, it is recognized as one of the most sophisticated self-regulatory systems in biology, coordinating metabolism, immunity, and longevity. At its essence, autophagy is a lysosome-mediated degradation pathway that allows cells to dismantle and recycle their own components. The process begins with the formation of an isolation membrane, or phagophore, that expands and encloses damaged proteins or organelles. This structure matures into an autophagosome, which later fuses with a lysosome to form an autolysosome, the site where cargo is degraded into amino acids, lipids and sugars, feeding the cell's renewal cycle. The machinery behind this process is exquisitely organized. The ULK1 complex initiates phagophore formation when mTORC1, the nutrient-sensing kinase, is inhibited under starvation. Beaclin-1, in association with VPS34, drives membrane nucleation, while the conjugation systems ATG5, ATG12 and LC3-PE coordinate vesicle elongation and closure. The lipidated form of LC3 decorates the autophagosomal membrane, a molecular signature now used as the gold standard marker of autophagy. Regulation of autophagy is tightly coupled to the cell's energetic state. The kinase AMPK, activated by an increase in the AMP-ATP ratio, promotes autophagy through mTORC1 inhibition and direct phosphorylation of ULK1. Meanwhile, the transcription factor TFEB governs lysosomal biogenesis, ensuring the degradative capacity matches metabolic demand. Together, these systems form a dynamic network, a metabolic thermostat maintaining homeostasis during stress. Yet, autophagy's purpose extends beyond nutrient recycling. It is a quality control mechanism, constantly surveying intracellular structures for signs of damage. Through selective forms, such as mitophagy, pexophagy and phagy, cells maintain organellar integrity. In neurons, where longevity and precision are vital, these selective processes prevent the build-up of defective mitochondria and misfolded proteins that can disrupt synaptic signaling. Defects in autophagy are now recognized as a molecular hallmark of numerous diseases. In the heart, inadequate autophagy accelerates mitochondrial dysfunction and oxidative injury during ischemia-reperfusion events. In metabolic tissues, impaired autophagy contributes to insulin resistance and beta cell failure. In the brain, the decline of autophagic flux underlies the accumulation of amyloid beta and alpha-synuclein, driving neurodegenerative cascades. Paradoxically, in cancer, autophagy plays both guardian and traitor, suppressing tumorigenesis in early stages while supporting established tumors under metabolic stress. This duality reveals a profound truth. Autophagy is not inherently good or bad. It is context-dependent, serving as a molecular switch between protection and destruction. In homeostasis, it preserves function. In pathology, it can fuel survival of the unfit. The boundary between adaptation and disease lies in the precision of its control. In the heart, where energy demand is relentless, autophagy functions as a molecular safety valve. During ischemia, it removes dysfunctional mitochondria to preserve ATP production and limit oxidative injury. When blood flow returns, however, excessive autophagy can trigger autophagic cell death, aggravating tissue loss. The balance between protective and destructive autophagy is governed by AMPK activation, BCLIN-1 regulation, and mTOR suppression. Pharmacological modulation of these pathways, using AMPK agonists, resveratrol or rapamycin analogues, has shown cardioprotective effects in experimental models, underscoring the therapeutic potential of tuning this delicate process. Metabolic diseases reveal a similar pattern of imbalance. In type 2 diabetes mellitus, pancreatic beta cells rely on autophagy to remove damaged organelles and unfolded proteins, ensuring insulin secretion remains stable. Chronic nutrient overload and oxidative stress suppress autophagic flux, leading to beta cell exhaustion. In peripheral tissues, impaired autophagy contributes to insulin resistance by allowing lipid intermediates and damaged mitochondria to accumulate. Conversely, reactivating autophagy restores metabolic flexibility, enhancing glucose uptake, reducing ER stress, and improving insulin sensitivity. In the nervous system, the stakes are even higher. Neurons are irreplaceable. Once lost, they are seldom regenerated. Autophagy ensures their longevity by clearing toxic aggregates and maintaining synaptic integrity. In Alzheimer's disease, the failure of lysosomal clearance leads to the buildup of amyloid beta and tau aggregates. In Parkinson's, impaired mitophagy, often linked to mutations in PINK1 or Parkin, results in oxidative stress and dopaminergic neuron loss. Yet, uncontrolled activation of autophagy may induce autosis, a caspase-independent form of death. Thus, neuroprotection depends not on maximal autophagy, but on its rhythmic precision. Aging represents the gradual erosion of that precision. With time, autophagic efficiency declines, LC3 lipidation slows, BCLIN-1 expression drops, and lysosomal pH drifts from its optimal range. The accumulation of cellular debris, oxidized proteins, damaged mitochondria, and lipofuscin drives senescence. Activation of autophagy through caloric restriction, exercise, or compounds like spermidine and rapamycin rejuvenates the system, delaying functional decline. Studies across multiple model organisms, from yeast to primates, suggest that autophagy is a central determinant of lifespan, acting as the cell's timekeeper. In cancer, however, autophagy's dual nature becomes most apparent. At early stages, it suppresses tumor initiation by removing damaged DNA and maintaining genomic stability. In established tumors, the same machinery supports growth under metabolic and oxidative stress. Autophagy provides a steady supply of recycled nutrients, enhances mitochondrial quality, and promotes survival during hypoxia or chemotherapy. Genetic deletion of key autophagy genes increases genomic instability, but also sensitizes cancer cells to stress-induced death. Therapeutically, both inhibition and stimulation of autophagy are being explored, one to starve tumors, the other to induce lethal self-digestion. Inflammation ties autophagy to the immune system's rhythm. By clearing damaged mitochondria and limiting reactive oxygen species, autophagy prevents uncontrolled activation of inflammasomes and cytokine storms. Defective autophagy in macrophages amplifies IL-1 beta and IL-18 release, driving chronic inflammatory diseases such as Crohn's disease and rheumatoid arthritis. Conversely, appropriate autophagy maintains immune tolerance, supports antigen presentation, and ensures resolution after infection. In cancer, this dual role becomes complex. Autophagy can suppress inflammation to limit tumor initiation, but later shield tumor cells from immune destruction. Finally, in infection, the oldest battlefield of life, autophagy becomes an active weapon. Through xenophagy, cells selectively engulf bacteria, viruses, and parasites. Pathogens are tagged by ubiquitin, recognized by adapters like P62 and NDP52, and enclosed within autophagosomes for lysosomal degradation. Yet evolution is reciprocal. Listeria, Shigella, and some viruses have evolved mechanisms to escape or hijack this system. Even so, autophagy remains integral to innate immunity, regulating antigen presentation and shaping adaptive responses. Enhancing autophagy may thus represent a future strategy against chronic infections and emerging pathogens. Across these diverse contexts, autophagy reveals its defining feature: adaptability. It is not a single pathway, but a fluid continuum that adjusts to stress, nutrition, and age. In every cell, autophagy sustains a dialogue between preservation and renewal. When well tuned, it ensures resilience. When disrupted, it marks the beginning of decline. Ultimately, autophagy exemplifies a profound biological principle. Survival is not achieved by resisting change, but by mastering it. Cells endure not by hoarding, but by letting go, by transforming waste into resource, entropy into order. In that silent cycle of destruction and renewal lies the essence of life itself.